and plant stuff. I have 4 of those, yeah. So I just need the other ones back out there if you would just, oh, I'll take it.
All right, so if you have not given your plant dirt, Donovan would love your dirt.
Anybody else who has some? Thank you so much. Awesome. Somebody else? Anybody else? Anybody else?
I want to say yes, because it'll be important because some of us will have amnesia after the presentations because they're so traumatizing we might not remember what we did. So Madison, you're just waiting on a working script. So just to add to the stress level, we said add to it. Right. But I appreciate it. Keeping the scientific process going. Yes, sir.
Test, test. Test, test.
She might.
Okay, can we have the presenters from Green and Madison come up on the stage? No one? Oh, here they come. And guys, just a reminder about the microphones. If you hold it down here, it doesn't work. It needs to be up here, especially for your parents and everyone watching on YouTube. If it doesn't come out of here, it doesn't go to them.
Hello, I'm Edith French, and we are your Green and Madison class of 2025. We've been testing heavy metals on our Arabidopsis plants. Green has been testing nickel sulfate, and Madison has been testing zinc chloride. Industrial runoff is one of the major issues in today's society. Industrial runoff is one of the major issues in today's society. As you all know, when this occurs, plants are affected. If the APP6 gene has a positive effect on the heavy metal tolerance of these plants, then that makes it a stronger candidate for use in plants of economic significance. Green has been testing nickel sulfate, like I said. They've been taking an aqueous solution of this nickel sulfate with a concentration of 8 millimolars and administering 1 milliliter to their plants every Friday for 6 weeks. Madison has been taking an aqueous solution of zinc chloride, and the concentration of that is also 8 millimolars, and they administered that to their plants every other day for 2 weeks. Each time they administered it, they administered 25 milliliters. When the plants were bottom watered, the heavy metals were pulled through the soil.
Hi, I'm Mariano Perez, and I will be presenting Madison's hypothesis. Our hypothesis is the Arabidopsis plants with the AAP6 gene still intact would outperform those that had the AAP6 gene removed when heavy metals are applied.
Hello everyone, I'm Riley Shiflett, and I will be discussing Greene County's hypotheses for their Arabidopsis study. On the slideshow, you'll be able to see the hypotheses for clarification. We predicted that the plants missing the APP6 gene would suffer when heavy metals were administered.
Hello everyone, my name is Isabella Strickland, and today I'm going to be presenting Green's results. Starting with the line graph showing the average rosette diameter, you can see that all of the plants had a steady increase and decrease. The photos on the top help support the increase in the graph and show what the plants look like at the beginning in the top left and through the middle of the experiment as seen in the top right picture. The box plot chart shows that there was no statistically significant difference between the wild type plants. However, there was a slight statistical difference between the mutant control and mutant experimental. On this next slide, you can see that the mutant control, seen in blue, had no bolt growth. On the other hand, the mutant experimental, seen in yellow, had the highest bolt growth. The pictures support this by showing the qualitative data to help support the quantitative data. The box plot chart shows again that there is no difference between the wild-type plants. However, there was a large difference between the mutant experimental and mutant control plants.
Hello, my name is Kayla Schaller, and I will be discussing the presence of seed pods in the Arabidopsis thaliana plants. As you can see from the top left graph, only the wild-type plants reached the point in their reproductive cycle where they were able to produce seed pods, whereas, as you can see from the photographs, the mutant types did not at all. We had no seed pods from the mutant types. And as you can see from the bottom left graph, there was a statistically significant difference between each of the data points. Thank you.
Hi, my name is Jacob Polsky, and I'll be presenting the data from Madison County. To the right of the image, you will see two sets of photos of the plants, which were one group of plants that we tested during our experiment. On the left of the image, we have the mutant type control, the wild type control, the mutant type treatment, and the wild type treatment. The photos on the left were taken before any of the treatment or watering was administered to the plants. The photos on the right were the same 4 plants after the treatment and watering had been administered over a 2-week period. As you can see, the treatment plants grew much worse than the control plants, and most of the treatment plants ended up dying due to the fact of zinc poisoning. The line graph demonstrates this data, which holds the average growth rate for all of the plants in the experiment. The Watson and Whisker plot also demonstrates this data, having more accurate numbers and having standard deviation and standard error included. Due to the closeness of the numbers that we measured, we determined that this data did not support a hypothesis that when the AP16 was present, the plants that were administered the treatment of zinc chloride would perform better than the plants without the AP16.
Hello, I'm Heath Miller, and I'm going to be presenting the errors for this experiment. So the first of these was that while green used distilled water, Madison used tap water, which has other minerals that we do not want to test. It just added extra variables to the experiment. The next thing was that different groups and different people had different definitions of what plants were dead and alive. And so this could have skewed the data a little bit, because you don't want a plant coming back to life after it's already been pronounced dead by another student. Also, for this experiment, we wanted to test 1 anion and 2 cations. Unfortunately, with the chloride also being administered, we had 2 anions and 2 cations. So this also threw off the data a little bit and made it a little less accurate. The last thing was that green's plants were under constant 24-hour light the entire experiment. This was a huge limiting factor and really dwarfed what the nickel was doing to the plants at the same time. So that really messed up the results for green. And then Madison's plants started 3 weeks late because their first experiment failed due to like a malfunction in the lighting fixture.
Hello, my name is Amy Gens, and I am discussing Green's conclusions once I get the clicker to work. There we go. Green had 3 hypotheses, 2 of which were rejected and 1 was accepted. Our data is difficult to come to a definite conclusion with, as the extent of the effects of the continuous light that our plants were exposed to cannot truly be determined, as we don't have a control sample for that. Our first hypothesis was a comparison of the effects of the addition of heavy metals into the soil of our Arabidopsis plants for wild types. This hypothesis was rejected as there is, as demonstrated by the graphs, no statistically significant difference between the wild type experimental and the wild type control plants. Green's second hypothesis was a comparison of the effects of disabling the AP6 gene. We predicted that disabling the AP6 gene would harm the plants, and this hypothesis was accepted as demonstrated by the graphs. There is statistical significance in the difference between the rosette diameters and the bolt heights. The mutant control plants never bolted, so we do not have a comparison for those measurements. Green's third hypothesis was a combination of the first two, predicting that the addition of the nickel sulfate combined with the disabling of the APSIX gene would harm the plants. This hypothesis was rejected as, again, there was no statistical difference between the wild-type experimental and the wild-type control, and there is only statistical difference in the graph on your right that, or between the mutant control and the mutant plants and the wild-type plants. And again, there is statistical significance in the difference of bolt heights, but between wild-type and mutant, so we cannot support this third hypothesis either.
Hello, my name is Riley Gordon. I will be presenting Madison's conclusion. Madison's hypothesis of if the APSIX gene is present, then the wild type of Arabidopsis will have a higher growth rate over the mutant type when zinc is applied was supported by the box and whisker plot beside you. The red and blue represent the control groups, while the green and orange represent the experimental groups. In both control and experimental, the wild type of Arabidopsis experienced higher growth overall during our measurement period. We can conclude this to be because the APSIX gene was present. However, there was not enough data to tell us that the APSIX gene had any effect on heavy metal tolerance. We say this because the wild type of Arabidopsis in both control and experimental were already growing at a much faster and higher rate overall compared to the control group. More tests and studies should be run to further conclude our hypothesis of APSIX gene's tolerance to heavy metals.
Hello. I will be discussing the overall effects of heavy metals and the suppression of the APSIX gene on the growth and well-being of the Arabidopsis thaliana plants. A common consensus from both Madison and William Monroe is difficult to arrive at due to the extent of the experimental errors. The data collected by Madison is ongoing as the plants are not yet old enough to arrive at any definite conclusions, and the data collected by William Monroe is undermined by the size of the sample group, which diminished significantly due to a lot of the plants wilting. However, some conclusions can still be drawn from remaining surviving plants, such as the heavy metals, nickel sulfate and zinc chloride, negatively impacted the growth of the Arabidopsis thaliana plants. And the suppression of the APSIX gene did not improve the plant's ability to withstand the effects of the heavy metals, and in fact, decrease the plant's ability. Thank you.
Hi, my name is Alex Vandegraff. I'm here to present the real-life application of our experiment. The purpose of our experiment was to test if the disabling or non-disabling of the APSIX gene is selective advantage. The ultimate hope of our experiment is that the APSIX gene could be introduced into agricultural plants such as wheat. Before farmers would like to introduce this gene, they would want to know what problems may occur. The key for the APSIX gene is it must solve more problems than it creates. And in our experiment, it did just that. It did not leave the Arabidopsis vulnerable to zinc chloride or nickel sulfate. This is important since zinc chloride and nickel sulfate are common in heavy metal runoff. To be sure, though, we would like to retest this experiment countless times to make sure this outcome was not an outlier. I would first like to thank my Madison and Green colleagues for working so well together. Next, I would like to thank our teachers, Dr. Clements Miller and Ms. Dexter. Then I would like to thank Professor McDowell and Virginia Tech for helping us with this experiment. Here are citations. We'll take questions now.
I think working with another school was definitely definitely complicated as we didn't have a lot of time to collaborate with each other and like ask each other questions, and even throughout our experiments we weren't like in constant communication with each other. Like we didn't know that Madison's plants had to restart their experiment until like last week. So that was definitely, there was a little bit of stress with communications, I would say. And I think for like why it's beneficial, it's definitely, well, splitting up the work was nice, so not one person had to give the presentation. That was appreciated. And I think also it allowed us to test a different variety of variables, like our comparison of nickel and zinc. I don't think one school would have been able to do that. So I think it was nice to run both experiments at the same time and have enough people to run it for each, and working with two different schools gave us that ability to do that. Yes, Professor McDowell?
So, congratulations. Great job.
Thank you.
You guys were really well organized and super poised. I appreciate that. And I also appreciate how well you integrated the presentation. The last thing that I'd like to comment is that I noticed one result from your experiments that was clearly supported between the two schools, even under the different light levels, which gives me confidence that it's a real focus. I'm not going to say what it is right now because I'm curious to see if other schools report the same difference, and also if other people inside the group can pick up on it. But overall, it was a really useful presentation. Good work.
Thank you. I would say that maybe having a camera to watch your timer to make sure that it is functioning properly while you are not present to babysit it, that would help so you can make sure all your technology is working. I think maybe some more organized communication, like within groups, like as Heath mentioned, different people had different ideas of what plants could be measured and what measurements were viable for the data. So if that was set as a standard beforehand, I think that would definitely help with that. Are there any other questions?
So we need all of you to stand under the screen. So as opposed to where they were standing, you guys both sides stand under the screen. And the clicker is best, she said, to put on this side to reach the dongle. Yeah, whoever stands on this side.
Is this on? It is. Okay.
Hi, my name is Katie, and this is Dylan. And the topic of our presentation was to see what the effects of adding urea and water to the silenced versus unsilenced AAP6 gene would do to the Arabidopsis thaliana plant.
Urea nitrate was selected for this experiment because it is considered the most valuable component for plant growth. Plants with a higher concentration of nitrogen should have more foliage growth, allowing for higher photosynthetic rates, which should overall lead to more plant growth.
Block 2 measured bolt height, rosette width, and foliage color, while Block 4 measured bolt height, rosette width, and the amount of leaves each plant had.
Both Block's hypotheses appear on the screen, but I will be breaking down Block 4's into simpler terms. We hypothesized to see a size difference between the mutant type and wild type plants. We hypothesized that the wild type plants would have a larger bolt height and rosette diameter, but the mutant type plants would have more leaves. We also hypothesized that the experimental groups would start off smaller, but continue to grow and end up being larger than the control group plants.
Block 2's hypothesis is also going to be on the screen, and to break that into more simple terms, we hypothesized that the addition of urea and water would increase the bolt height, rosette width, and change the foliage color of all the plants, but would have the most significant effect in the AAP6 gene present plants.
Anna and Kara will now be explaining how our hypotheses compare to our results.
Okay, so displayed on the graph above are Block 2's averages for the rosette widths. And as you can see, most of the data points were very clustered together until the very end of the experiment, where a couple plants in the mutant type control group died, which caused the average to drop. There was a slight source of error in this because the dead plants were only recorded for 1 day, and then the next time that data was recorded, it was not recorded, so the data point goes down and then right back up. Also at the end of the experiment, the wild type experimental group experienced a jump in growth. This was most likely due to the presence of the AAP6 gene and the addition of 2.5 grams of urea. The wild type experimental group ended up with an average of 63 millimeters, and the other 3 groups ended up with averages between 52 and 55 millimeters. The data is in support of Block 2's hypothesis, which stated that the wild type groups would do better because of the AAP6 gene and that the experimental groups would do better because of the urea. This is, now Block 4's rosette width averages are displayed, and their results were a bit more definitive than Block 2's were. About halfway through the experiment, the wild type control group started growing more than the other groups, and at the end, the error bars were not even touching between the wild type control and the rest of the groups. The wild type control ended up with an average of 77 millimeters, and the other 3 groups had averages between 52 and 57 millimeters. The most plausible reason for this result was the presence of the AAP6 gene and the lack of urea. Since Block 4 added 10 grams of urea, it is most likely due to the fact that, the addition of 10 grams of urea was most likely too much for the plants and ended up being more harmful than beneficial. This is only in partial support of Block 4's hypothesis, which stated that the wild type groups would do better and the experimental groups would also do better, but the experimental groups were not as good as the control groups in the long term. And next, Kara will be presenting from home due to an illness.
Key with the dark color symbolizing block 2, and the lighter color symbolizing block 4. We're between the wild type and the mutant type of smaller average bolt height than the mutant, especially in the findings for second block, as you can see, they ended up at the bottom of the graph outside of this main trajectory, which all of the other plants followed. So, and block 4 as such is in this main trajectory, the green and the red right here. So the similar trajectory that we're talking about is this area at the beginning towards the end, where they all kind of fall into the same path and locale, and at the end they'll all fall into the place of about 60 to 80 millimeters. We especially see the similar trajectory in the mutant experimental of both classes, these yellows right here, and they follow nearly the exact same path throughout the entire experiment. We saw differences in the controls of the classes, most specifically in block 2, with the red and blue right here, where they landed on completely different places in the graph, supporting conclusions that AAP6 gene does affect growth. For differences in experimental, it was more minute. Again, in block 2, the yellow and green, and this may support conclusions that wild type and mutant type respond differently to treatment. The outliers of the main trajectory were the wild types of block 2, with the wild type control being the lowest on the graph, and the wild type experimental following just short. The most confusing point of data is right here on the wild type experimental on 1026, where it was very high and then dropped suddenly. This was most likely a calculation error. Overall, block 4 had a much higher average bolt height, most likely due to an increase of urea treatment. So second block was treated with 2.5 milligrams of urea in 1,000 milliliters of water, and block 4 was treated with 10 milligrams of urea in 10,000 milligrams of water. Block 4 also recorded the average amount of leaves, but since students calculated this manually and counted manually, these are not reliable data points, and such would be a source of error if presented. Block 4's hypothesis cannot be validated because there was no real difference in the growth of mutant type and wild type in rosette or bolt width. In rosette width and bolt height, my apologies. So here you'll see that they all fall into the same trajectory, all of the light colors are nearly in the same place. And here in rosette width, you'll see that for the most part, they all follow the same path, with the exception of which being wild type control, but since the wild type experimental did not follow that path, we cannot conclude there were many major differences. The bolt and the rosette were not larger than the wild type as expected in the hypothesis. Once again, this trajectory is nearly the same the entire time, with the exception of wild type control. And here everything is in the main trajectory, all of the light colors for block 4. Lastly, the hypothesis cannot be supported because the experimentals did not start off smaller in the beginning of the experiment. Here they are all in the same path, on the same up-down motion, same situation, and here following the same path in the very beginning. Thank you, and we'll now pass it on to conclusions.
Okay, so before we go to conclusions, the last point is that Block 2 recorded color of the plants, and there was seen to have a difference between the plants treated with urea and the plants that were not treated with urea. However, it was more just observable data rather than quantifiable data, and for that reason, it would be a source of error if put into a graph, so it is not being presented on a graph today.
Thank you, Anna and Kara. Before I start the conclusions, I would like to change a point made earlier within the presentation. Kara said that Block 4 used 10,000 milliliters of water, but instead they actually used 2,000 milliliters of water. But as you can see, based on our data, 2.5 grams of urea did not have a significant effect, whereas 10 grams of urea showed more pronounced results within the bolt growth and color of the plants.
The purpose of a bolt height is to disperse seeds around a plant. Now, there was lagging in the experimental groups, but this is not necessarily a sign of malnutrition. In fact, in the control groups, the quick bolt growth is a sign of premature death. This is due to if a plant is about to die, it will extend its bolt in hopes of reproduction. Now, this could probably be due to the absence of foliage growth that the urea provided to the experimental groups.
The plants being treated with urea showed a much darker hue as well. This is likely because of an increased production of chlorophyll B, which tends to have a darker hue than chlorophyll A, but there's no quantifiable data to support this claim. If the experiment were to be conducted again, it would be most useful to use amounts of urea between 7 and 15 grams.
This experimentation is beneficial to plants in many areas. Though we used artificial conditions, a natural environment has many downsides, such as parasitic nematodes. The absence of the AAP6 gene has been proven to aid in this presence. Now, more experimentation with parasitic nematodes would be beneficial to plants for its results.
Furthermore, if the experiment were to be if these two variables were used together at the right amounts, farmers could use them to decrease crop loss and fight off pathogens in environmentally safe and efficient ways.
Finally, our data has proven that urea will help foliage growth, which is very beneficial to plants because it will help utilize sunlight.
And as you can see in our picture, this is a picture of the finished product of plants in Block 2's class, which had the lower amount of nitrogen use. We would like to thank Mr. McDowell, Mr. Connor, and the BRVGS staff for providing us with the materials necessary to conduct this experiment.
Thank you for listening, and we are now open for questions.
On the far, that is the mutant type control. Next to it is the wild type control. Then it goes to the wild type experimental and finishes off with the mutant type experimental.
Thank you.
Yes.
So, first thing I'd like to say is thanks to you guys and everyone else in Atlanta for hosting this event. I know that's a big deal. I appreciate that. Second is that, again, you did a terrific job on the presentation. I'd like to hear a little bit more about your color observations. I appreciate that you were, conservative about discussing that observation because you can't quantify it. But qualitative data is okay too. Okay? Often people present that just by showing examples of differences. So tell me a little more.
Yes, so as you know, second block used a lower amount of nitrogen, and we were the ones that measured color, but there wasn't really too much of a difference in color from second block's plants, but fourth block, which the increased amount of urea, they showed more of difference of colors and darker hues, which I said earlier is probably an increased production of chlorophyll B, but we don't know for sure. Yeah, that's about it. Yeah, the plants were a bit darker green. We had a color chart for block 2, and they were mostly sheen green, which is probably a shade no one really knows, but that is the color that we recorded week in and week out when we measured.
Yeah. We did think about that, and it was decided that they would be recorded, and I think that just when the data was recorded the second time, like after they died, I think people just forgot to add them in or just didn't add them, so that was why there was a big change in the data.
After discussion with a few people, we decided that those amounts would be best because we wanted a lower amount and a higher amount just to see if a lower amount would do the job and if a higher amount, if that may kill the plants. So we made sure to try by research to see if we could lower it just to the right amount.
So I think what she was trying to say was that the wild type experimental group, it had a quicker production of the bolt height, but at the end there were probably some deaths or maybe a calculation error. So that was why it went up very high and then down immediately after.
As well, there was a 2-week or 1-week period, I believe, when the plants were not treated because of a weird scheduling at school, so this could have also had an effect on our data and our graphs.
What I also wanted to say is that in our group, we actually had an incident where we dropped a pot. Okay, yes. We did. We aerated the pot. And then after that, we replanted the plants, and one of the plants did die. Another error we had that assisted in the drop on the graph is that we did have some surface level mold. It was not on the plants, but that could still be apart from dropping on the floor and maybe some dirty hands.
We believe the mold is from certain groups eating over the plants, maybe once, as well as particles within the air of breathing on plants, and the dial calipers, sometimes when people would be measuring them, would touch the plants with their hands, which could have caused mold, but not as many groups as I thought had mold as they did. Yeah. Yeah. Some groups had it worse than others, but overall it was pretty good.
Yes.
So the way that we did it, the urea was placed into the solution of water, and then they were bottom watered into the treatment box. We just did the exact measurements. We put 2.5 grams within 1,000 milliliters, and then 10 grams within 2,000 milliliters. We thought that would be best, as if we put 10 grams in 1,000 milliliters, that could have led to like oversaturation or too much, and so we thought that the 10 grams in the 2,000 would have been better.
Any more questions? Okay, thank you for your time.
idea. Who's gonna start?
Hi. Hello. Okay. Hi, we are Gooch High School, and we're going to be presenting on the effects of acid rain on Arabidopsis. I'm Kaylin. This is Abby. That's Malachi, Sam, and Will.
So just a little bit more about us. The way our class was split up, me and Kaylin were in block 4 together, and Malachi, Sam, and Will were in block 3. Now, although we both tested the effects on acid rain, we did it with different pHs. So the majority of our class did pH 4 and pH 3, and the majority of their class did pH 5. And a little bit about acid rain, just where it comes from. Acid rain comes from manufacturing, power plants, fossil fuels, basically anything that emits these acidic chemicals into the atmosphere. Volcanoes and actually rotting vegetables can also emit these acidic chemicals into the atmosphere. From there, the rain it collects in the clouds and the rain will come down. Now, acid rain has a pH around 4.2 to 4.4, and in comparison, normal rain is about 5.6. So it's a pretty big change compared to what we usually get. A little bit about our experiment. We used the pHs of 3, 4, and 5 on our experiment on the Arabidopsis plants.
We hypothesized that as the pH of the acid rain decreases, so will the growth of the plants, as well as that the mutant plants will exceed the growth of the wild-type plants.
The purpose of our experiment. So as both of our classes decided that it would be a good idea to take a real world issue that is going on, like a real form of pollution, and we chose acid rain. Because we wanted to see, since we have two types of plants, how the different pHs would affect the mutant plant and the wild type differently, since the mutant plant has a gene that has been disabled.
So to begin, I'll be discussing our control group of Arabidopsis plants. In any experiment, the control group is a group that does not receive any external modifications. It's used as a baseline for the experiment and gives us a point where we can compare our data once it has been collected for the final time at the end of our experiment. In this experiment, the control group was made up of wild-type and mutant-type plants. These plants were not given any acid rain solution. These plants were given fresh water whenever the experimental group was given its acid rain solution. We hypothesized that the control group would grow much better than the experimental group because the control group's plants were not under any kind of external stress.
We tested with the experimental groups. We tested with the pH of 3, 4, and 5. We started on September 28th and ended on October 30th. We tested with the same amount of solution, same pH solution on all the plants, and that was independent from the AAP6 gene. And yep, that's all I got to say.
This graph shows the growth over time of the bolt height of the control group of plants. The red line represents the wild-type plants, while the blue line represents the mutant plants. We noticed signs of stress showing on October 21st when the mutant plants began to show redness around the rosette, slight shriveling in the leaves, and a decrease in growth. This is contradictory to the wild-type plants, who showed a pretty consistent growth throughout the entire testing period until October 28th, when both plants seemed to reach their end of their life cycle. This seems to show that the additive of the deactivation of the AAP6 gene did not improve the life cycle of the mutant plants too much, except for a slight growth spurt in the very beginning, which consisted through the entire thing until October 21st, where their growth decreased dramatically compared to the wild-type plants. Similar to the control group, the pH 3 plants showed very similar signs of stress at a very similar time, with redness around the rosettes, decreased growth, and very limited shriveling around the rosette. However, this occurred between both the mutant and wild-type plants, not just the mutant.
The mutant and wild-type plants in this group showed many growth patterns. The plants grew at very similar rates. However, it's important to note that the mutant plant started out much larger than the wild-type plant. This is due to the mutant plant growing significantly in the first few days of the experiment when data was not originally being collected. The AAP6 gene did not seem to have much of an effect on the plants being grown with the pH 3 acid rain.
Signs of stress that we observed with the pH 4 plants. We saw that the pH 4 plants had much, or the mutant pH 4 plants had much earlier bolting than the constant wild types did. So we observed, since this bolting is a sign of stress, just because it's the reproductive system of the plant starting to happen, and we just noticed that this happened a little bit earlier than expected with the mutant plant, but overall wasn't too detrimental to our experiment.
So how does the AAP6 gene fit into our experiment? The AAP6 gene is the intake of amino acids, but the mutant plant actually had this gene deactivated. So that is another reason why we chose to do acid rain, is because we wanted to see how the different pHs would affect those different types of plants with the gene and without the gene.
Some of the things we saw with the mutant versus the wild type was just with the control variables, just treating the mutant and the wild type with water. We still observed that the mutant plant observed to be fuller, brighter in color, and taller in height, whereas the wild type still looked like it was growing at a steady pace, but just not as extravagant as the mutant plant was.
So in the end, we were actually surprised that the strength of the pH 4 was actually harming the plants a little bit more than the rest of them. Like you can see, compared to pH 3, it'll show up. pH 3, our data is a little bit more lower than the pH 3 sooner in the process because, yeah.
So this is the pH 5 data group. As you can see, they kind of started at the beginning. You see the slight increase with the yellow on that, or the experimental mutant, and we saw that the AAP6 gene was the reason that we saw that slight increase at the beginning. But on October 21st, both plants started to sign stress, but continued to grow until October 30th, where they both died off completely and shriveled up.
So the results that we collected prove that the AAP6 gene did not have much of an effect on the survivability or productivity of Arabidopsis plants affected by acid rain. The graphs that we created clearly showed that the mutant-type plant was always larger than the wild-type plant. It must be noted, though, that the plants grew at very similar rates, and the mutant-type plant simply started out as larger than the wild-type plant. Our graphs also showed that there was not a significant change in the growth or productivity of the plants based on the pH they were exposed to. pHs 3, 4, and 5 were all very similar and showed very subtle differences. Finally, our control data was very similar to our experimental data. However, it must be noted that the wild-type plant did surpass the mutant-type plant in our control data. We believe that because the AAP6 gene was disabled in the mutant-type plant, that it may have shortened the plant's lifespan.
The overall goal of our experiment was to see whether or not the AAP6 gene would show positive or negative effects when compared to non-AAP6 gene disabled counterparts when acid rain was introduced to their system.
the overall goal of our experiment. So, pretty much the overall goal was to see if acid rain was going to help or harm the plants. And in the end, no, it did not really help the plants at all. So we do not think that acid rain should be used in crops, such as potatoes, corn, because it didn't really help very much. Okay. What could be improved? Basically, our class started 1 week late than the timeline that was given to us. And because of that, we struggled a little bit in the end when we were working with the meat of the project. It's important as scientists to stick to the deadline that you are given, and also to have a consistent data collection, especially because we had 2 classes. We struggled a little bit because we did not have a set in stone consistent data collection, so all of our graphs looked a little bit different. Finally, more research before the project actually started. It's important to know what you're doing before you actually get into it. And with this experiment, we found out that we were a little bit lost because we had not done the research before to know the correct amount of stuff to actually do our experiment. Citations and questions and concerns. Any questions? Yes. Pardon? Oh, we did height, width of the plant, and the rosettes. Oh, no, the bolting. Bolts, rosettes, and leaf distance. The diameter of the leaves. Yes. Yes, our data did make it to the seed pods, but a mistake that we made was we stopped collecting the rosette length and we started doing different sets of data and put those all in our graph instead of keeping them separately. So we have, in our graphs, we have information for rosette diameter and the seed pods. So it's a little bit of both. Yes. I think it really depends with our.
We did see that acid rain did harm the plants, but we also observed that if the AAP6 gene was disabled in the mutant plant, or was the mutant plant with the AAP6 gene, it did have a little bit more resilience with the acid rain than if we had just treated the wild type with acid rain. So although the acid rain was not good for a plant, I mean, it's acid rain, it's not going to be helpful to anything, but we did see that with the mutant plant, it did stand a little bit more of a chance than the wild type. Well, when we were treating the mutant plants with the acid rain, we saw that not only did they bolt sooner, but they had a little bit more fullness to them, whereas, like, this is kind of the word I'm looking for, but the wild type plants looked a little scrawny. Like they weren't very full or thick, or just, they didn't look as healthy. They just kind of looked kind of in the middle, in between. But the mutant plants did show a little bit more of a growth and a little bit more of a that's what I'm looking for. They just looked better. They just did better.
As we're trying to say, the mutant plants, they went through the life cycle more in a full capacity, while the wild types kind of just went a little bit and then just kind of stopped growing and stopped like completing the cycle, and they kind of just dropped off. Any other? Yes. Yes, we did, and I think we would have also used box plots if our data had been a little bit more clear, because as we were saying, we kind of stopped collecting like data. We didn't have a full set of data for each type. Like we didn't have a full set of data for the rosette diameter, and we didn't have a full set of data for the bolting. So if we had had a full set of data for both of those, I think it would have been more beneficial to use a box plot. But we kind of just had to use what we had. Yes. That's a good question. So with the actual pH water, we did not mimic the acid rain. We just kind of poured it on there, 25 milliliters, poured it on there every Friday. But during the week, we did bottom water them with normal water. So the experimentals and the controls were being bottom watered every day. But on Fridays, we took only the experimentals out and just did a normal little dousing of the 25 milliliters of the acid rain mimicry. Yes. Yes, so with the experimentals, with watering them so much, we did find that the solution did kind of just run through. I don't think that it had enough time to settle into the soil and have a good effect. So maybe if we had watered all of the plants less, we would have had better results and maybe more clear results, because like we said, the pH solution kind of just fell through the plant. It didn't really like sink in. Yes. Yes, in the back. Okay.
It was similar to like a brownish maroon color. They didn't turn bright red, but they did show a sign of deteriorating with like a dark maroon color. We did see the wild type change a little bit faster than the mutant did. So they got a little bit. Yeah, we didn't really see the mutants do it that much in general. Like towards the end of their cycle, they still looked like they were a healthy cycle, and except for the wild types that kind of slowly started to get that red color. Yes, I'm sorry. Yes. Mm-hmm. Yes. Yes, sorry. They bolted a little bit, but they were substantially smaller bolts than the mutant plant had. If anything, they didn't really bolt that much.
Yes. So with the difference between the wild types and the mutants considering bolting is that the mutants, they kind of just bolted, like the records like of growth bolting was like kind of off the charts for us, while with the wild types, they didn't fully bolt. They kind of just, like we said, they were growing, and they kind of just stopped. Like the bolting stopped. Like all progress moving forward with those plants, including the bolting and the seed pods, kind of just fell off. Like we were still collecting new bolt and seed pod growth with the mutants, while the wild types we were collecting less. It was kind of like the wild types were starting to deteriorate and starting to just fall off while they were.
Hey guys, we found a cell phone, so if you are missing a cell phone, let us know. Yes, otherwise it goes to the front office at Fluvanna.
guys weigh in that way. You're almost going to be standing together. Yep. Bring it on in. Snuggle up. All right, so you guys, the block split here? Yeah. Okay, microphone for each side.
Here.
So we're Orange County, and we studied the effects of various salts and the AAP6 gene mutation on Arabidopsis plants. And that is first block with Alex, Allison, Catherine, and Rachel. And this is third block with Adam, Dylan, Liam, Faith, and Michael.
So over the course of the 40-day period, we tested two variables, magnesium sulfate and sodium chloride. Magnesium sulfate is primarily used in mining, medication, and bath salts, while sodium chloride, which is commonly known as table salt, is used in road dethawing and medication. Now, the primary difference between these two variables is the chemical composition, considering the fact that sodium chloride is made of sodium and chlorine, and magnesium sulfate actually comes from a naturally occurring mineral called epsomite, which consists of sulfur, oxygen, and magnesium. Now, the reason that we're using these variables is because road runoff is an extremely prominent issue in today's society when it comes to agriculture. And as is analyzed, we have both of these salts extremely common in the runoff that affects these plants. And the Center of Agriculture, Food, and Environment has stated that generally salt is detrimental to most plants. Therefore, we've decided to analyze the effects of these salts on plants since they are so common in runoff.
In this experiment, in the control groups, we watered them using distilled water, while the experimental group was watered with using these two salts diluted in 1 liter of water, 4 grams in each one. The hypothesis for first block, the magnesium sulfate group, was that the wild-type groups would grow more than the mutant groups because the wild-type groups had the enabled AP6 gene, while the mutant-type control groups and experimental groups, they have the disabled AP6 gene. And since the wild-type groups proved to be beneficial because of the AP6 gene, we concluded that the wild-type groups would grow more than the mutant groups. While we also predicted that the experimental groups would grow more because magnesium sulfate is actually Epsom salt, which is commonly used in gardening to help with plant growth. So our prediction was that the wild-type experimental would grow the most, while the mutant control group would grow the least.
The third block hypothesized that the experimental groups would grow the least because sodium chloride has been proven to have detrimental effects on plants. In addition, the plants that have the AAP6 that aren't mutated, they would also grow most because they can have nutrients transported throughout the plant.
In this picture, you can see where on the left is the plants when they were around when they were first planted and when they started to germinate, and then on the right is when they started to grow their first leaves. This is the graph for the average rosette diameters for the class testing magnesium sulfate. And as you can see in this graph, the wild-type plants grew at a faster rate than the mutant plants. which is because since the AB6 gene is present in the wild-type plants, the amino acids are held in the leaves of the plant, which causes more structures to be built, which causes bigger diameters. And in this graph, there's a pretty steady growth up until the 18th, where the control groups started growing at a faster rate than the experimental groups. And then on the 20th, all 4 groups started to decline in diameter size. And in this graph, the experimental groups grew at a lower rate than the control groups, which shows us that the absence of the variable magnesium sulfate was more beneficial for the growth of the rosette diameters than the presence of it. And in these pictures, you can see on the left side, the wild-type plants had larger rosettes than the right side, the mutants had.
Above me is the box plot for the group that treated the plants with magnesium sulfate. And as you can see, in general, the wild types had higher measurements for the rosettes compared to the mutants. And there is a lot of overlap in this graph here, so there was no significant difference between the plants.
This is the graph for the sodium chloride tested plants. And as you can see, from September 30th up through October 14th, all of the plants grew at a pretty steady and equal rate. And then at October 14th, they sort of spread out in their growth patterns, and the wild type control really starts to grow over all of the other plants, which makes sense in theory, considering that it has its AB6 gene and it's getting fed just distilled water. So you would think in the beginning that that would make the most sense. And this is a picture of some of the rosettes, and as you can see, the wild type control, has much larger rosettes than the rest of them.
This is a box plot of the Arabidopsis plants, the average rosette diameter. So in this graph, you can see that the wild type group had the biggest or largest average rosette diameter, and that the mutant experimental type had the smallest average rosette diameter, while it was close to the wild type experiment. However, these graphs are pretty close in size, meaning that there isn't really any big significant data.
The next chart above me is block 1's bolt height graph, which they treated the plants with magnesium sulfate. And as you can see, the bolts started to grow on October 11th, and they grew pretty consistently until October 14th, when the mutant plants started to grow taller bolts at a faster rate. And this could be because of the absence of the AB6 gene, because they don't need it as much because they have smaller leaves. The nutrients can go throughout the plant and make them go through the life cycle faster, giving them taller bolts. And then as you can see here, the experimental plants did better than the plants, which could indicate that the salt did help the plants grow taller bolts. And then in this picture, you can see on the left, the mutant plants had significantly taller bolts than the wild type.
This box plot shows that the mutant plants had a much higher average in bolt heights than the experimental plants, especially the mutant experimental. But since there still is overlap in the box plot, there's no significant difference in the data.
This is a graph for the average bolt height of the plants tested with sodium chloride. Our first bolts began to grow on October the 8th, with the mutant plants growing before the wild type plants. The wild type plants got their first bolts around October the 11th. This could be because of the absence of AAP6, which could allow the mutant type plants to grow quicker and go through their life cycle faster, which gives them taller bolts and more time to grow. This can be used in the real world by farmers and crops to allow them to grow faster and produce more. This is a picture of our plants on grow day 38, and you can see the mutant type plants have taller bolts.
This is our box plot graph of the average bolt height for our sodium chloride groups. It should be noticed here that the mutant experimental type had the tallest average bolt height. The wild type experimental, the other experimental, had the smallest average bolt height. Both of the control groups grew a little bit separated here in the average bolt heights, as the mutant control grew much taller than the wild type control. However, all of this data, They are all overlapping, meaning that there isn't really much big significant data.
This bar graph shows the average Salique number for each plant, and as you can see, the mutant plants had a lot more Saliques than the wild-type plants, and this means that they had a better fitness and will have more reproduction, so that in future generations, there'll be more mutant plants grown than wild-type. And between the controlled and experimental groups, the experimental groups had more Saliques. And in these pictures, you can see some of the Saliques on our plants.
Above me is the box plot for the plants treated with magnesium sulfate, and as you can see, all of the plants except for the mutant control had smaller box plots, which indicates when the Saliques were collected, they had similar amounts on each plant. However, in the mutant control, there was a large variety and range in the data between how many Saliques were collected on each plant.
This graph shows the average number of Saliques for the plants tested with sodium chloride, and much like the plants tested with magnesium sulfate, the mutant plants had much higher average numbers of Saliques, and we think that this is because the AP6 gene not being in place will allow the nutrients to spread more freely throughout the plant, and it requires less energy to do so, meaning that you would get more Saliques on average. And this is a picture of some of the Saliques on the bolts on one of the last grow days. And as you can see, for the mutant plants, there are very tall bolts and there are a lot of Saliques on each bolt.
This here is a box graph of the amount of Saliques through our Arabidopsis plants. The mutant experimental type had a noticeably larger amount of Saliques compared to the other ones, especially the wild type experimental, which had the lowest amount of Saliques. It should also be noticed that the mutant control and the wild type control had a pretty similar amount of Saliques, but the mutant control was a little bit larger in the amount of Saliques. Though there is overlap in this data, you can see the mutant experimental had a much larger amount of Saliques, but really this is insignificant because of all the overlap.
Some main sources of error that could have happened during our experiment was the watering of the plants and how we measured the rosettes and the bolts. It took us a couple tries to figure out exactly how long we wanted the plants to sit in a certain amount of water. And then when measuring the rosettes and bolts, we could have missed a rosette or missed a new bolt height. And then, so there was lots of room for human and operator error. And then one thing though that we thought could improve this experiment is the further application and testing of the salt on the plants, which could give us different data to draw more conclusions on how exactly the salt affected the plants.
So one notable piece of first block's data was actually our Salique numbers. So our wild type Saliques were quite low in number, while our mutant Saliques were quite high in number, especially our mutant experimental plant, which had the highest number of Saliques overall. However, since the mutant control plant was only about 1 Salique lower on average, we would say that the effect of magnesium sulfate on the AP6 gene of Arabidopsis plants is inconclusive.
And then another anomaly in the data for third block is also the amount of Saliques on the plants, in that the experimental plants tended to have a higher number of Saliques on average, which is contrary to what would be expected because they were the ones that are exposed to the sodium chloride. And sodium chloride has been proven to be detrimental to plants, so it would be expected for it to follow suit with the Salique count. However, it seems that it helps the reproduction of the plants. However, because the data is so close together, there is no, it's not conclusive.
Now, while most of the data was insignificant, there are a few data points I'd like to look at for real-world applications. First being the removal of AP6 having an increased rate of reproduction plant. This can be used by farmers for plants such as corn that require higher reproduction rates for better yields. Other data point I'd like to look at is the detrimental effects of the sodium chloride. This can be used by farmers and environmentalists to petition the city to find better ways to treat the roads during the winter other than just laying a bunch of salt down. And this is our works cited. we'd like to give a special thanks to Professor McDowell for the information he gave us at the beginning of this, and Ms. DeVise for helping us through the entire process. Thank you.
Any questions?
Yeah, so I said that because all the way up until the end of the experiment, the mutant plants were actually greener in color, and we couldn't really put that on a graph, and it wasn't super significant enough to really talk about it, but they were slightly darker greener, and they were slightly darker shade of green, my bad, and that usually means that they were a little healthier, so that's why. In the end, the plants that weren't treated, I mean, all the plants sort of looked the same at the end. They were all like dying, but the color for the untreated plants was a lighter shade of green. A couple of the rosettes were starting to like brown up a little bit at the ends, but the mutant plants still kept a darker, more vibrant shade of green, even though they were starting to wilt. That was the average number of siliques per plant. So, yeah. Yeah, it was all just an average.
Yes. Yeah.
The salts were 4 grams and diluted in 1 liter of water.
So throughout the experiment, we noticed the growth patterns with the salt and non-salt. But overall, if with an increased concentration, I think it would be more harmful to the plants than beneficial. So some salt is actually good for plants, but in a very minuscule amount. But if we had increased the salt amount, I do feel like the plants would have wilted a lot faster or not grown as well.
Just to add on to that, personally, it was, as mentioned before, it was a really big surprise that the plants with the sodium chloride specifically seemed to actually grow decently well. But as that is believed to be an anomaly, if that sort of concentration were to be increased, the chances of that anomaly happening again would likely decrease. So if the concentration was increased, we believe that the plant would probably die faster due to dehydration since salt absorbs water.
Yes. It was bottom water.
notice if they're holding it down here or moving their head around, you can't hear them. So you almost want to hold it right in front and make sure you should be able to hear yourself over that. If you don't, it's not picking it up as well. And if you turn to look up there, take the microphone with you, okay? Who's doing the clicker? You guys? Tim? Okay. All right, so just make sure you're under the screen.
Hello. Hello.
This way? You guys?
Yeah.
There you go. And as I told them, you notice if people are holding it here, you can't really hear them. And if you look up, take the microphone with you. You should be able to hear yourself coming out of there. All right? And then who wants to do... Okay.
You want me to introduce y'all? I don't know why I got volunteered. No sweat. No sweat. I'm not. I am not sweating. It's good. I'm so nervous. Also, good push. All right, we got this. We got this. I don't think I can do much stress, honestly. Hi, my name is Landon Borman. This is Addison Smith and Jackson Claypack, and this is the effects of potassium chloride on Arabidopsis thaliana. We are from Louisa.
We are from Nelson. My name is Jasper Cooper Lake. This is Matthew Castillo, Katie Mars, and Nathan Manthe.
So, our background information. Arabidopsis thaliana is a model organism for genetics because it has been tested many different times, where this allows the DNA sequencing to be studied thoroughly, and which allows us to collect more data on Arabidopsis thaliana when research are performed. It also has rapid cycles, which means it has 8-week life cycle, and each week has a different type of growth and development, which also can be studied and collected, and is genetically transformed, which is done in this experiment with AP6.
There is a gene of particular interest to scientists present in Arabidopsis thaliana. This gene is called amino acid perme 6, or AP6 for short. Its role in plants is to help with transporting amino acids, you guessed it, throughout the vascular tissues of the organism, especially the xylem. Now you might wonder, why would a mutated version of this plant, which deactivates the gene, possibly be helpful? Due to the nature of certain microbial pathogens that live off of stealing the nutrients being transported through this process, they will starve because they are unable to get the nutrients. So, one of the things this experiment is trying to look at is, can the mutated plants compete with the experimental plants?
Okay, so we chose the variable potassium chloride, and potassium chloride is a common chemical component found in fertilizer around the world.
To administer the variable, Louisa made a solution of 0.02 grams dissolved in 1,600 milliliters of water, while Nelson made a solution of 0.04 grams dissolved in 1,600 milliliters of water. And potassium chloride is used as a common ingredient in many fertilizers as it's associated with the transport of water, nutrients, and carbohydrates in the plant.
The hypothesis for Louisa is if 0.0003 moles of potassium chloride was added to Arabidopsis thaliana, then there would show a 10% decrease in bolt height and rosette width in the wild-type experimental, and a 25% decrease in rosette width and bolt height in the mutant experimental when compared to the wild-type control.
As a class, Nelson decided that our hypothesis would be if potassium chloride is added to Arabidopsis thaliana, then there will be a statistically significant difference between rosette width and bolt height compared to the control plants.
Our materials and methods, so we administered the variable in a 0.02 gram solution in 1,600 milliliters, and each bin, experimental and control, got 800 milliliters of water, but the experimental got 800 milliliters of the potassium chloride water. They were also bottom watered.
Nelson administered the variable the same way, using a 0.04 gram administer of potassium chloride in a 1,600 milliliter solution. It was split up into two 800 sets and given on each Tuesday and Thursday of every new week. Some possible experiments, or some possible errors that we ran into during our experiment were that in early stages of development, group 1 did drop their wild-type experimental plant, causing to a setback in growth. We struggled with measuring the same plant every single time due to bad spacing of the plants, which made it hard to make sure they were measuring the same one every time. And our last error was that our hypothesis for Nelson was made very vague, and that led to a lot of gray area and inconsistencies when it came to drawing a conclusion for our experiment.
So Louisa also had a few errors, which our group 1 dropped their mutant experimental while measuring and in the middle of the experiment. And then group 4 placed their wild-type control group into the experimental bin, which then absorbed some of the potassium chloride that was in that bin overnight.
These are Louisa and Nelson's wild-type plants. So the wild-type plants have the AP6 gene activated. So this gives them that gene in their DNA. The plants up here, the front view shows the wild-type control with, you can see the bolts that are coming up. And then you have some front views and top views of the wild-type experimental and the wild-type control.
On the left over here, you've got Nelson's wild-type plants. The one on the left shows a front view of Nelson's wild-type control, while the two on the right show a view from above of the wild-type control and the wild-type experimental plants.
Here are Nelson and Louisa's mutant plants. On the left is Louisa's plants. These photographs were taken on the last day of measuring. They are side view pictures and display the bolt height.
On the right over here are Nelson's mutant plants. On the left you've got their mutant control, and on the right you've got their mutant experimental. What makes the plants a mutant plant is that the AP6 gene is essentially deactivated.
This is Louisa's graph on rosette width. As you can see in the graph, the wild-type plants had a higher overall average in rosette width than the mutant plants, and this indicates that potassium chloride did not really have an effect on the plants, but rather the activation or deactivation of the AP6 gene cause the plants to have either a higher or a lower average in rosette width.
This is Louise's box plot graph. As you can see, the wild type graphs have a larger range in their growth, and the mutant groups have a smaller range, which indicates that wild type did grow healthier when in rosette width, while mutant did not grow as great at their rosette width.
In terms of Nelson's rosette width, we initially saw the wild type control experience the most growth. It is also important to note that as of this part in the experiment, both of our experimental groups are just a touch behind the control groups in terms of their development. This box and whiskers plot is valuable because it shows that at this point in time there is very little statistical significant difference in the gathered data. This is apparent by the whisker overlap that shows the quantitative overlap of our information.
This is Louisa's graph for bolt height. As you can see, the mutant plants did much better in their growth cycle. For bolts, they were much taller. The control variants grew more than the experimental variants. And as you can see, the wild types pick up like at a random, and that is because we had not measured the first bolt since 1018.
This is another box plot for Louisa that shows the bolt height for all of our types of plants. And this one, compared to the other one, the mutants did significantly better in bolt height than rosette width, and we think this because of AP6 amino acid transporting, so it may have transported more to height than in the width.
This is Nelson's graph, line graph representing bolt height. As you can see in this graph, the wild type control ends up being the tallest bolt that we have, although all of the plants show a similar growth rate and growth path. There is not much difference between any of these, leading us to believe that they all look very similar, which they do, and are qualitative data. This is Nelson's box plot for bolt height. This graph shows, as well as our original box plot, that there is little to no statistical significance because of the overlapping of the whiskers throughout all of our plants.
This is Louisa's select count for the Arabidopsis plants. As you can see, the mutant control and the mutant experimental had the highest amount of selects recorded. The wild type plants had little to no selects recorded. This is because as the bolts mature, they develop seed pods, otherwise known as selects, and these were counted much higher in the mutant plants than they were in the wild type plants.
This is the graph for Nelson. It shows the effect of potassium chloride on the number of selects produced by the mutant and wild type plants. Selects are long, narrow seed pods that are produced in the final life stages of a plant. As shown on the graph, the mutant control plant had the highest number of selects, and the control groups had a slight higher count than the experimental plants.
In conclusion, for Louisa, we found that potassium chloride had no difference in the rosette growth or the bolt height of the Arabidopsis plants. However, the AP6 gene did allow for the plants to have differences. In the wild type plants, they showed a higher average rosette width in the first stage of their life, and then for the mutant plants, they showed a higher bolt height at the end stages of their life and a higher select count. This leads to the observation that the AP6 gene starts and ends the life cycle earlier than it should, depending on if it's activated or deactivated.
Nelson concluded that in this experiment, the addition of potassium chloride had no statistical significant effect on our plants, either the plants that were given potassium chloride or not given potassium chloride, and there was also no difference in the mutant and the mutant and the wild type plants that we had. This was displayed as all of our graphs had very similar growth rates and growth patterns, and our box plots also displayed very similar data. A real-world application of this potassium chloride is that it's used in many fertilizers, as Matt stated earlier, and this information can be used by farmers to determine how much potassium chloride or chloride in general to put into their fertilizers. Any questions? Yes, sir. As far as I know, there was no significance of the specific number that we chose. We just wanted a low variable group and a high variable group, so we had the 0.02 and 0.04, but there was no specific measurement, as far as I know, based on fertilizers that were used.
Yeah, so for Louisa, we did extensive research to find what experiments had been done already on potassium chloride to see what values that were already known of to hurt the plant or to help the plant, and we found some middle ground there to find a lower value or to find a higher value to see if it had any effect otherwise on the plant. Yeah, so we looked for a long time and we did not find much extensive research at all on potassium chloride that had been tested on plants in general. So I think we might have found one or two sources that had a specific value that showed that it caused the plant to die. So we went a little bit lower than that to be able to find, not necessarily kill the plants, but to see necessarily how it worked against the plants.
Yes. At least from Nelson's data and our conclusion, we think that the amount of potassium chloride that we added was not enough to affect it, and I think given more, it would have a large effect on it, obviously killing some of the plants. And like Louisa said, they did do research on like a general area that has been tested of potassium chloride already on plants, and we just went below that as to not kill the plants, but to still get information on it. Now, Nelson did not come up with a conclusion that changed what we knew, or we just kind of assumed that it wasn't enough to affect the plant.
Louisa didn't notice much of a color trend based on the two groups. I think it was just the AP6 gene that changed whether the plant used all their energy to make bolts or whether it was rosette width in the beginning of the life cycle.
I think to properly answer the question, we would have to conduct more experiments to really find out whether it was the concentration or the AP6 gene that played the largest role in determining that. We did collect sleek numbers at the very end of the experiment. I believe we did it twice. We had 2 days for data collection.
I think it was actually, or something that we noticed when doing graphs near the end of our experiment was that some groups
had done averages of the saliques over a certain amount of time, and then other groups had just done the flat number of only one day that they measured it. So I think there was also some inconsistency there.
Yes, that's correct. I think that the wild type would get to the same amount of saliques that the mutants had, but I don't think there would be a time where the mutant plants would have the same amount as the wild type plants.
Even at this point in the experiment, a lot of our plants were already beginning to sort of wrap up their life and begin to die. So I don't really think they would have enough time to have grown any more saliques. It's always really interesting to see what someone who's doing something similar to you is able to come up with. It was a really interesting experience getting to work with another school.
Yeah, so I think it was really important to be able to have our own, like Louisa have their own group and be able to test just one thing and not have the classroom divided between two different variables or two different amounts of the variable, and then when we're done to put all of our results together and say, okay, Louisa had this, Nelson had this, and we got to see the real differences between. And I feel like if we just had it in the same class with the same people, just the different amounts of the variable, then we wouldn't have had the same visuals that we had getting from Nelson.
Okay, any more questions? All right, thank you.
Hey guys, I do want to draw your attention to something. I know most of you probably aren't on Facebook and it's just your parents and grandparents, but I still share with that for our social media, and I heard just 2 days ago when I sent out the link for this for people to be able to watch, and this is a comment I got very quickly. This is a OCHS from 2005. So she did this experiment in 2003, almost 20 years ago, and that it changed the trajectory of her life and her career with this project. And you may be thinking, we didn't get any statistically relevant data, it didn't show anything, but when you combine that with 20 years of students researching this, that becomes very powerful to Virginia Tech in terms of what they know. Here is a little, the second comment. I messaged her immediately because I wanted to know more about where she went with this, and I thought it was really neat that she took this and got 2 degrees in the topic from Virginia Tech, and then has gone on to have a career with Monsanto and one of the Bayer subsidies that does technology and agriculture. So this may seem like a project that you're just so thankful is almost over, but this could really be something that if you're interested in it moving forward. She lives in Unionville in Orange, and she offered to come speak. If when you're a senior you want to do something with this for your project, she would be more than happy to reach out and help anyone just on this topic moving forward. So just is a lot bigger than just you guys today, that this is 20 years